Protein synthesis is a result of transcription and translation. The process starts when DNA is transcribed into an mRNA strand. The mRNA strand is then read and translated into a polypeptide chain in the ribosomes. The transcription of DNA is carried out by the enzyme RNA polymerase. This enzyme reads one of the DNA strands in a double helix and creates an RNA strand that corresponds with the sequence that was read. DNA and RNA pairs match as follows: A with U, T with A, and G with C. In a DNA strand, the standard base pairs are adenine, A, and thymine, T, cytosine, C, and guanine, G. The base pair U is not present in DNA, but while a strand of DNA is being transcribed, the mRNA strand will have uracil in the sequence as the corresponding base to adenine. In DNA, there are two strands, the template strand and the copy strand. The copy being all the complementary pairs of the template. When DNA is transcribed into mRNA, it becomes the same as the copy strand except thymine is replaced with uracil. In this strand, the base pairs make codons in groups of 3. These determine what amino acid is made. In translation, the mRNA strand is fed through the ribosome, which helps to assemble the amino acids. Amino acids chain together to form a polypeptide chain, which is the basis of a protein. Most codons code for a specific amino acid, but one codes to stop the process and cut off the chain. This is known as a stop codon. Inside the ribosome, tRNA creates anticodons, which are the complementary bases to the codons. When the codons match with their respective anticodon, the ribosome begins to make the amino acid. After the amino acid is created, another anticodon takes the previous one's place. mRNA is separated into groups of 3. These codons often code for the same amino acid because code is redundant. Most amino acids have multiple codons that translate into it. This is important because it is the reason many mutations do not have an effect on your body, and it is known as a wobble. In review, transcription takes the template strand of DNA and transcribes it into RNA. This can be thought of as writing down something that says the same thing in a different manner. For example, writing a paper from research. It is the same information but presented differently. You are transcribing the information. Translation is where mRNA is made into a protein. This can be thought of like in a language class where you're taking one language and translating it into the one that gives you a grade for the class. In the same manner, the protein is what is really important, but it must be taken from the RNA first.